generations, and Gwen got trained as a phlebotomist in order to be able to do this study. What we don't understand is when we look at all of these results that everyone is presenting, all these brain scans, all of this cortisol stuff, all the spit, all the hair, all the urine, all the blood, all the cheek cells, that doesn't happen without people like Gwen. She will go into these houses. She can get blood from a turnip. Okay? They have seen mice, they have seen roaches, they have seen lice. They will stay at these houses for 5, 6, 7 hours. She is traveling all over the United States. Some days she's in Texas and then in New York. And I just want to thank you from the bottom of my heart. You are wonderful. Thank you. And it's her birthday. But it's a reminder that our teams are important, and these people are hard to find, and they're harder to keep. And these longitudinal samples don't come back if they don't have a good experience with the people who go in. So thank your coordinators. Hug your coordinators. Pay them well, all right? And let's not forget on whose back this research is built, okay? So thank you so much. With that, I'm going to turn it over to Chad Schenk, another of our network faculty, and we'll get started on some very exciting sessions. Thanks, Chad.
Thank you, Jenny. So we have an excellent session devoted entirely to the topic of genomics today. And instead of listening to me for any longer, I'm going to jump right in and start to introduce our first speaker, Kieran O'Donnell. Dr. O'Donnell received his PhD in clinical medicine at Imperial College London under the tutelage of Yvette Glover. He also holds a master's degree in neuroscience from the Institute of Psychiatry at King's College London. He is currently a CFER postdoctoral fellow in child and brain development at McGill University, where he works primarily with Dr. Michael Meany. Dr. O'Donnell's work examines how early life factors influence the function of the genome in the developing child. His research takes place in the context of a number of prospective longitudinal cohort studies where he characterizes patterns of DNA methylation across the genome using array-based platforms. These collaborative studies examine how early adversity becomes biologically embedded and the implications of such effects for child mental health. Dr. O'Donnell's work seeks to identify epigenetic signatures of positive early life experiences, such as early interventions, that buffer some of the effects of early life stress. This has particular relevance to the field of child maltreatment. He is really leading the way in how early adversity, such as maltreatment, leads to sustained changes in the methylome that increase the risk for psychopathology. By studying some of the positive effects of early life experiences, he's able to spearhead translational efforts, specifically in the area of maternal sensitivity, that can help inform prevention models on the adverse effects of child maltreatment through more effective epigenetic programming. With that, I'd like to introduce Dr. Kieran O'Donnell.
Thank you very much, Chad. That was a very comprehensive welcome. I'd like to start by thanking Jenny and Sandy and all of the team here at Penn State who have been incredibly welcoming. And having experienced the ice cream from the creamery yesterday, I now know why you call it Happy Valley. Let me just exit out of this. So today I'm going to tell you a little bit about the research that I've been doing as part of my postdoc working with Michael Meany at the Douglas Research Hospital in McGill. And like you, my research is very much focused on the role of the early environment in shaping child development. And I'm particularly interested in the mediating molecular mechanisms that link early experience to later mental health. Now, yesterday, Bruce Ellis gave us a wonderful introduction to the idea of the developmental origins of health and disease. And a core feature of that is the idea of developmental plasticity, the idea that you can have multiple phenotypes arising from a common genome depending on the context in which that genome finds itself. Now, I find that idea really interesting because it's something that's conserved across a wide range of species, from lower insects to higher order mammals. We see a profound influence of the early environment and various different developmental processes. So for example, in the meadow vole, we know that the thickness of its fur and the timing of its sexual maturation is influenced by the level of maternal melatonin during gestation, which in turn is influenced by the length of daylight that that female experiences during gestation. And of course, that changes with the changing seasons. Likewise, in the red squirrel, we know that females exposed to high levels of population overcrowding during gestation give rise to offspring that show accelerated weight gain during the first year of life. And we know that these effects of overcrowding on offspring weight gain are mediated, at least in part, by maternal glucocorticoids, as you can recapitulate this phenotype by spiking in glucocorticoids to the food pellets that these pregnant females eat. And then, as we heard yesterday, there's really quite a large number of studies now showing a profound influence of maternal care in the rat, whether it be with regard to immune function, obesity, or anxiety-like behavior. We see marked differences in the developmental trajectories of those individuals exposed to high or low levels of maternal care. But we know that these effects are not just restricted to rodent models or preclinical models, but are also observed in human populations. So drawing on the work from my work with Tom O'Connor and Vivek Glover during my PhD, we used the ALSPAC cohort, which is a large epidemiological sample of 15,000 women and their children that have been followed longitudinally from early pregnancy until the present day, when these children are now having children of their own. We made use of their data on child emotional and behavioral difficulties collected from 4 to 13 years of age. And what I think you can appreciate from this graph is that those children born to women with high levels of prenatal anxiety, represented in the dotted line, show elevated symptoms of emotional and behavioral difficulties at age 4. And these effects really do persist until at least age 13. And because this is a large epidemiological sample, we can control for a whole host of potential confounding factors. But what I think you can appreciate across each of these examples, from the meadow vole to the women and their children from ALSPAC, is that there's a central importance of maternal signals. That maternal signals are conveying information about the early environment and influencing offspring outcome. But the question then becomes, what are the potential biological mechanisms that are sensitive to these maternal signals, that are mediating these effects of the early environment on child outcome? So in the last number of years, there's been growing interest in the role of the epigenome. Now, when I talk about the epigenome, I'm talking about a series of chemical marks or modifications, either directly to the DNA or to the proteins which DNA is wrapped around, the so-called histone proteins, which you can see represented in blue here. Now, I think it's really quite an amazing feat when you think about it, because in each and every cell in our body, if we were to stretch the DNA out from end to end, there would be approximately 2 meters of DNA in every cell. And what the epigenome allows us to do is to package this DNA in a really effective manner. So the parts of the DNA that are necessary for that particular cell at any given time are accessible to the transcriptional machinery that needs to read the DNA, and the rest of it can kind of be compacted away and packed away nicely. Now, the particular epigenetic modification that I'm interested in is that of DNA methylation, which is the addition of a methyl group, so a chemical modifications, directly to cytosines in DNA, so that's the C in the DNA sequence. Now, why are we interested in DNA methylation? Well, conventionally, DNA methylation has been thought of as a repressive marker or a repressive function. And that's partly due to its association with functions like X inactivation, where you have the silencing of an X chromosome in XX females. But we also know that DNA methylation is important in gene silencing in the function of imprinting, for example, where you have the silencing of a maternal or a paternal copy of a gene. So you can see why DNA methylation has a history with gene silencing. But what I think is emerging from the literature on DNA methylation is that it can have more nuanced effects on gene expression. And rather than being a simple on-off switch, it's a bit more like a dimmer. So it's kind of regulating the amount of gene expression depending upon the needs placed upon that cell at any given time. But one of the other aspects of DNA methylation that I think is particularly interesting is that there's growing evidence that DNA methylation is also involved in a function called alternative splicing. Now, this is the mechanism whereby the same section of DNA can give rise to subtle differences or subtle variants in the message that's produced, so-called splice variants. So we're beginning to understand that DNA methylation is not just regulating the amount of message that's produced, but also the type of message that's produced. So this is quite important because it allows our static genome, a single genome, to generate a diverse number of transcriptional programs, depending on the needs placed upon that cell. So going back to DNA methylation and its function on gene expression, how does that happen? Well, we know that methylated cytosines are less likely to be bound by certain transcription factors. So those proteins, when they can't bind to the DNA, are less likely to activate the transcriptional program and less likely to mediate gene expression. But we also know that DNA methylation can recruit repressor complexes. And this can influence the 3D structure of DNA. So you remember I was talking to you about the compaction of the DNA? Well, this repressor complex can mean that that DNA is more compacted and less likely to be accessible to the transcriptional machinery and less likely for that gene to be expressed. And as you all know now, over the last 10 to 15 years, there's been a growing number of studies showing that early adversity and early adverse circumstances can influence variation in methylation across a number of candidate genes, with one of the first examples being that of IGF-2 in conjunction with exposure during the perinatal period to the Dutch hunger winter, which was a brief period of famine in the Netherlands. We also know from work with Rachel Yehood and, of course, Michael Meany, that we see variation in the methylation of the glucocorticoid receptor as a function of parental Holocaust exposure, but also, and I think perhaps most relevant to this audience, following exposure to child maltreatment. So these studies historically have used a candidate gene approach, but more and more we're turning to genome-wide methods to try and assess variation across the genome, or across the methylome, some DNA methylation across the genome. So what are the tools that we have at our disposal to do this? Well, the platform that I've used extensively during my postdoc has been that of the Illumina 450K. Now, this is a microarray-based platform, which many of you will have heard the term microarray previously in terms of gene expression, and this is assessing DNA methylation. And this provides quantitative data on DNA methylation at single nucleotide resolution at just over 480,000 methylation sites across the genome. Now, while that sounds like a huge number of sites, it's actually just a fraction, less than 2% of the 28 million CPGs in the human methylome. But I think what's nice about the 450K is that rather than just focusing on the promoter region of the gene, which has historically been a region that we've all been very excited about, it gives us information about DNA methylation across the gene and over different genic features, moving from the promoter region across the gene body down to the 3' UTR. And here you're seeing what you typically get from the profile of a gene with the 450K. On average, you have around 17 CPGs per gene, but that will depend on the size of the gene. And you can see lower levels of DNA methylation in the promoter and then higher levels of DNA methylation in the gene body. But the analysis of the 450K is something that is evolving. And it's something that we're constantly trying to think about how we can do it better. So after measuring DNA methylation at over 480,000 sites, the first thing we try to do is reduce the number of sites that we look at. We try to figure out which are the sites that are going to be most informative. So the first strategy that we do is we only look at methylation sites that show inter-individual variation. So if these sites aren't variable, then we don't include them in our analyses. So one of the first things we could do is, for example, linear regression analysis, represented here on the lower left-hand corner with the volcano plot, showing the effect size on the x-axis and the p-value on the y-axis. The next form of analysis that we can do with 450K data is principal component analysis, where we're reducing these variable probes, so that can usually be around 145,000, down to discrete independent components. So maybe the first 10 principal components, for example. And we look for an association with our phenotypes of interest. And here what we're asking is whether or not our predictors of interest are influencing variability, or influencing the variance that we see in our DNA methylation data. And the last form of analysis that we've been using in the lab more recently is called weighted gene co-expression network analysis. So this is a network-based approach to methylation data analysis, where you're trying to identify those sites that are correlated with one another, that may be forming a functional network together. So the first study that I'd like to tell you about is using data from a longitudinal cohort in Montreal called the MAVEN cohort. And here we're focusing on the role of the early environment in shaping the methylome. And for this particular study, we're focusing on infant attachment, which we heard yesterday was also related to two aspects of neuroimaging outcomes. So for the purposes of this study, we conducted the strange situation at 36 months of age, and we grouped the children in terms of their attachment security, so secure, insecure, and disorganized. And as you know, the attachment classification is kind of thought of as a readout of the infant's perception of the early caregiving environment, with a sensitive classification being more likely when the child's needs are met in a responsive and a sensitive manner. Oh, sorry. I'll go back to that one. So we had a cohort of 108 children, and we classified them into the different attachment classifications. And the first question that we asked was whether or not we would see any association between infant attachment and child mental health. So we had symptoms of child emotional and behavioral difficulties at age 7 years of age. And what we found was a significant prediction of child mental health as a function of infant attachment. So we saw significantly elevated symptoms of emotional and behavioral difficulties in terms of total behavioral difficulties. And also when we looked at the subscales that were affected, we saw that these were particularly pronounced for inattention and hyperactivity disorder. And I think what you can appreciate from this graph is that it really seems to be the disorganized group that's driving this association. So the next question that we asked was whether or not we would see any association between variation in DNA methylation. So we ran the 450K using these buccal epithelial cells, so these cheek swabs from these kids, and we ran a principal component analysis on the variable sites, the variable CPGs, and what we found was a significant association between infant attachment at 36 months of age and DNA methylation, variation in DNA methylation at 7 years of age. And I think what you can appreciate from this graph is, again, it seems to be the disorganized group that is showing the strongest association with principal component 2, which is accounting for approximately 7% of the variance in DNA methylation. So then the question that we had was, we see an association between infant attachment and variation in DNA methylation, and we see an association between infant attachment and child mental health. Is there any overlap between the sites that are associated with infant attachment and the sites that are associated with child mental health? And what we found after running a permutation analysis was that there was approximately 900 CPGs that showed a significant overlap between infant attachment and child mental health. And what was potentially very interesting about this is that this seemed to be enriched for synaptic function, so processes related to synaptic function. So although these are results that certainly require replication in a larger sample, and that's something that we're actively pursuing in the lab at the moment, I think these results provide proof of principle that infant attachment in the early care environment associates with methylome variability, and that where these sites overlap, they may show a prediction with child mental health. Now, I think these effects are particularly interesting when placed in the context of early intervention programs that target mother-infant interactions and try to improve the early care environment and thus improve child outcome. So the question then becomes whether or not early care or early interventions that focus on mother-infant interactions in the early care environment would also influence variation in DNA methylation and the relevance of such effects for adult mental health. And that was a question that we set out to directly test in collaboration with David Oles, who many of you know has devised the nurse-family partnership. And we were making use of the randomized control trial in Elmira, which was the first site for the randomized control trial of the nurse-family partnership. Now, as you know, the nurse-family partnership is a perinatal parenting intervention, and it targets high-risk women, so teenage mothers in their first pregnancy, usually from low SES backgrounds. And we know that it's focusing on three principal domains. One of those is maternal lifestyle factors, so trying to reduce smoking and drinking, giving dietary advice. Also focusing on maternal-infant interaction and practical childcare advice. And we also know that the third domain is focusing on maternal development. So trying to get these women to complete their education, and we know that one of the program outcomes has been rejoining the workforce. And of course, giving advice on family planning, with again another program outcome being an increased inter-pregnancy interval. But perhaps one of the outcomes from the nurse-family partnership that is most relevant to this audience is there was a significant effect on child maltreatment. So David and his colleagues published this, showing that the nurse-visited group had significantly less child abuse and neglect up until the age of 15. So the question that we had was whether or not we would see any association between the early intervention program and variation in DNA methylation. So David and his team were able to send their team to collect blood samples from participants in the nurse-family partnership. Now these are children born to women in nurse-visited or control groups that are aged 27 years of age. So this is 25 years post-intervention, because the intervention starts in approximately mid-pregnancy and nurse visits continue until around 2 years of age. So obviously there's a long intervening time between the termination of the program and the assessment of the blood, and we'll come back to this in the limitations of the study. But the first question was whether or not we would see any association between treatment and variation in DNA methylation. And indeed we did. So we saw a significant but a fairly subtle association between early intervention treatment group and variation in DNA methylation. So I do want to highlight that this is a principal component that's accounting for approximately 1% of the variance in DNA methylation. So it's not a whopping effect, but also bear in mind that we've collected these blood samples 25 years post-intervention. So I think it's kind of interesting that we see this association at all, especially when we control for various different confounding factors, including, and importantly including, cell composition of whole blood, as we know that you can have tissue-specific effects on variation in DNA methylation. So all of the results that I'm showing you here are controlled for ethnicity and cell type. But what we did observe in this cohort was that it was really quite a marked effect of a history of child abuse and neglect on variation in DNA methylation. So we found an association between child abuse and neglect and multiple principal components. But one of the things that we did notice about this was that there was a significant effect of lifestyle factors. So when we ran our linear regression analyses to identify those CPGs, those sites that were most strongly associated with a history of child abuse and neglect, we found that many of these effects were mediated by increased risk-taking behaviors in those children that were exposed to abuse and neglect, including smoking, for example, with significantly higher rates of smoking in those individuals with a history of child abuse and neglect. But having accounted for all of those effects, so cell type, smoking, gender, ethnicity, we identified approximately 11,200 CPGs that showed an association with child abuse and neglect, independent of all of those various different confounding factors. And we asked the question, well, is there anything interesting about these CPGs? Is there anything that, kind of tells us more about where these CPGs are or what they may be involved in? And we ran an enrichment analysis, and we found that these CPGs were located in or very close to transcription factor binding sites, particularly for the glucocorticoid receptor, the estrogen receptor, and the progesterone receptor. Now, this was particularly interesting to us because the idea of steroid-mediated remodeling of the methylome is a mechanism that's gaining increasing evidence to support its role in influencing or mediating the association between the early environment and methylome variation and potentially later mental health. So as we heard from Christina Heim yesterday, Elizabeth Binder and her group have been focusing on FKBP5 demethylation as a function of early trauma exposure. And we know that this occurs within a glucocorticoid response element, whereby the binding of the glucocorticoid receptor to its consensus binding site, to the GRE, is influencing the level of methylation at that site, reducing the level of methylation at that site. This mechanism has also been shown for the estrogen receptor, and then some excellent functional work from Dirk Schubler has shown that this also associates with other transcription factors such as CTCF and REST1. Now, it's not every transcription factor that when it binds will influence DNA methylation, but particularly for the glucocorticoid receptor and these steroid hormone receptors, there seems to be evidence that the binding of its